Wonderful. Thank you. Perfect. So first, just want to say hi, everyone, and thank you for joining today's testing event. So we're very excited to have you here and looking forward to getting started. We very much appreciate your attendance and participation today. The DataX core team and the Lantana team have done a lot of work to put this event, as well as the draft IG together, and more work to come. But you're playing a very significant role here in providing us with this very crucial feedback on the IG's usability, even at this early stage. So this is a major milestone for us, and we're very glad to have your support. So a big thank you to everyone here today. With that being said, we have a pretty full agenda. So I have a couple of intro slides here that are prepped just to go through to kick us off. So first, let's just take a quick look at today's agenda. Not sure if I'll need the full 30 minutes for the introduction. Just have a couple slides just to get us started and provide some info on the day. And then I'll hand over to Sheetal Gaigi, who will introduce the testing scenario. And then Rick Smithies will take us through an overview of the HTML rendering of the FHIR output. And he'll do a live demonstration of that, FHIR data exchange process. And then at around 10 a.m., we'll take a 15-minute break. And then that will conclude the more sort of presentation-focused aspects of the testing event. And the rest will really depend on active discussion and participation from everyone. So when we come back from the break, we'll do an initial discussion on the technical demonstration, any comments or feedback related to what's presented at that point. And then following that, we'll hold some time for any general discussion on any other aspects of the testing event or the IG. And then at the very end, we've saved a little bit of time to talk about the FHIR test server. Okay, just a couple housekeeping items before I get into the rest of the introduction content. So first up, just wanted to address that all the information shared during today's meeting should be presumed to be public. So if you're submitting any information to the FHIR test server, please just make sure it doesn't contain any proprietary or confidential information. We'll share the link to that FHIR test server at the end of today's call. So if you want to spend some time trying it out and using it to exchange any sample data, we certainly welcome that. And please feel free to provide us with any feedback that comes from your experimentation. Please just note that this is a test server, so this is not the server that you will eventually see and interact with as part of the Accumulus platform. This test server was stood up as an environment for today's event specifically, so just keep that in mind. And we can provide some very basic tech support if you're having issues with the server, but we wouldn't be able to provide very detailed training on how specifically to use that FHIR server at this time outside of what will be presented in the demo. So you might need a little bit of technical expertise to be able to use that server. Yeah, detailed training is out of scope. So I mentioned this a little bit earlier when I was going through the agenda, but just wanted to kind of re-emphasize that we really want to hear from you during this testing event. So this is meant to be sort of a collaborative working space for us. We really want this IG to be, an industry-led effort, and for this to be usable within all of our companies and beyond. So we need all the constructive comments, questions, and feedback that we can get. In a way, we're all designing this sort of future vision for CMC data exchange together. So please don't hold back any thoughts that arise today, and both technical and CMC related feedback is very welcome. And as you may notice, the earlier portions of today's discussion will be recorded, and those will be uploaded to Box and Teams just for your future reference. But after the break, we'll stop the recording. We won't record the discussion portions, but we will take some detailed notes and then share a high-level output with the team after the testing event has concluded. Okay. So you've most likely seen this slide before in some of our testing event materials, but just wanted to level set where we are in this process of developing the IG. So this is our first testing event with this IG, and we'll have at least two more following this one, planning currently for July, and then to attend the September public testing event at the FHIR Connect-a-thon. So since this is our first testing event, we're hoping to take a lot of lessons learned from today's event to bring forward into those future testing events. And then the other thing that I wanted to note here on this slide is that we're ultimately working towards an April 2024 publication date for the IG. We're expecting to have some synergy with other Accumulus platform related timelines and deliverables. So that's ultimately, the date that we're working towards. Okay, and you most likely have seen this slide as well, but just, making sure that we're level setting and all in the same place with today. So this is the overview that was originally shared with the CMC subcommittee and tech committees to request your participation today. But sort of the purpose of today's event is to start testing the international CMC FHIR standard, which we're calling DXPQ, that will ultimately drive the DataX platform. So we're looking for feedback on the implementation guide and the FHIR profiles that will be used. Would be very helpful to, from a CMC perspective, to start identifying any gaps, misalignments, and fits for the use case. So, for example, we're going to be using, these FHIR profiles for exchange of CMC data for today, we're going to be looking at a variety of different domains, perhaps most notably stability. So if you are a CMC expert and you notice particular gaps in information that you would typically provide in a CMC application in the stability section, please let us know about that. We want to make sure, that the IG is as complete as possible so that this is a usable IG for us for exchanging regulatory information. And we've highlighted a few initial focus areas here. I think on the next slide, I'll talk about the different waves of how we're organizing, tackling all of these different domain areas. But we've selected a couple that we've prioritized because we feel that these are, really high impact, high value, and areas that we wanted to tackle first. So we have 14 different PQ domains that will ultimately integrate as part of the IG. And so what we've done is we've kind of split these into three different waves just to allow us to, have sufficient time to cover all of these areas. So we're currently in wave one at the moment. So this is our wave one testing event. So the domain areas that are in scope for today's testing event are listed here. Stability is a primary focus area, but we will also cover specification, batch analyses, DS and DP identification, which is, nomenclature, organization, which is really the manufacturing facility information. There's also some overlap with impurities, analytical procedures, and container closure system, because there are some elements of these three sections that appear in the stability, specification, and batch analyses sections. So we'll see a little bit of overlap there, but we won't have all of the details for those three sections filled in at this point, just what we need to support the current testing scenario. And then after today's testing event, we'll start working towards wave two and working towards another testing event in July. So we'll take the feedback that we get from today's meeting to improve those wave one areas, and then also bring whatever feedback is relevant into wave two as well. So in wave two, we'll start looking at manufacturing process validation, and then we'll finish up all of the remaining details from impurities, analytical procedures, and the container closure system. And then in wave three, we're really looking to complete all of the domain areas that we've identified. So there's DSDP characterization, batch formula, DP composition, compatibility, and reference standards. And that will lead us up to that public testing event in September, the Connect-a-thon. So we should have all of the domain areas filled out at that time. And of course, this is iterative, so we'll take all of the feedback that we get from all of the testing events and work that into the IG, preparing for publication. So this aspect was addressed in the sort of overview that we distributed in advance of the April 11th meeting. Just kind of wanted to go over this quickly. So we offered sort of two kinds of participation options. And of course, this will all run simultaneously today. So we have participants from group one and group two all working together in today's meeting and having discussion together. But we know that there's a mix of expertise in the crowd. We have CMC experts who are going to be in our participant group one that will be able to provide us with, feedback on the PQ content, product quality content. Um, so we would really depend on group one to provide us, kind of that gap assessment and that domain expertise relating to the applicability of the standard to regulatory requirements and the CMC domain. And then for group two, this is the more technically focused sort of track. And we gave participant group two the option to edit the XML files that we had provided in advance of this meeting so that they could use those on the test server that we will provide a link to and kind of go through that sort of exchange paradigm and provide feedback on that. So we'd appreciate, any feedback on the sort of technical process from group two. And a couple things that are out of scope for today's testing event. So I think infrastructure, any comments or questions relating to setting up a FHIR server and APIs, probably out of scope for today's discussion. Though with Accumulus standing up a FHIR server as part of the platform, I would imagine that there would be sort of an outlet for this type of discussion moving forward. Um, PQ domains. So we'll mostly focus on with the wave one topics that I showed. So things like manufacturing process validation, impurities, the detailed aspects of analytical procedures, container closure system, anything outside of the stability related aspects, batch formula. Those are all out of scope for today's discussion, but we will see those coming into play in the next couple of months. And a quick note on modalities. So the sample data is broadly scoped for a solid oral dosage form. That's the area that we have sort of started developing in. It's sort of a straightforward example, but other modalities are out of scope. So there might be missing elements in the sample data that you might see, for example, in a biologics application. Um, we're open to feedback on other modalities, but that won't be sort of the focus for today. But definitely we will need to prioritize extrapolating the IG elements to include other modalities. So we do welcome this type of feedback, but we're not hoping to focus on that today. And then any operational aspects of the testing scenario. So for example, we set up a testing scenario where there is kind of a data exchange happening between party A and party B, and who specifically party A and party B are. We won't go into details discussing that, whether party A and party B are, the applicant or a CMO or a health authority. We won't really be discussing that. So we're just kind of trying to demonstrate data exchange from, place one to place two. Okay. And I believe this is my last slide here. So about 30 minutes ago, I did send an email to the full attendee group, which contained a feedback form. So we provided this template just for all the participants to fill out. So I sent that via email attached, but it's also in Box and Teams if you're able to access those shared spaces. And this template is really, just for collating any comments or feedback that you might have. And we have some guidance in the template itself regarding, how it should be filled out. And optionally, giving you the option to either fill these out individually as a participant or by company if you wanted to kind of collate all the feedback by company. It's really up to you. However is easiest for your organization to provide this feedback. And then we're requesting that you please send your completed forms back to me. And there's my contact information there. And please send those at your earliest convenience after the testing event. So we'll have a look at these and compile all the feedback, and this will be important for us to target making changes, and we'll go by the priority level. So I think that's everything from me for the intro. I'll just ask my colleagues quickly. Anything else to add? Any other kind of level setting we should do before we move on?
No, nothing from me. I think you covered it.
Yeah, all good for me, Rita.
Same here, Rita.
All right. Thanks, Mike, Craig, Sheetal. So I will pass things over to Sheetal now, and I will stop sharing if I can do that.
So I just started sharing. Can you see my screen?
Yes.
Yes, we do, Sheetal.
All right, perfect. So what I would be going through today is providing the overview of our testing scenario, and the focus, as Rita mentioned, being on stability. But before going into the actual stability section, just wanted to bring up the concept, how we are developing these sections, and what Rita a couple times mentioned, that everything is either like connected, and there are linkages. So something may be out of scope for the details, but we might be connecting or requiring just few data elements from some of the other sections. This is just a quick snapshot on a big big picture on the data components, say for a drug product in the CMC domain. So if we speak about the ECTD, the CTD structure, I'm referring to the product section, module 3 product section, if we will. And as noted here, this is the full drug product data domains, CMC. But that can be further divided into 4 like content blocks. It is just a hierarchical model. And in the content block, there can be the actual product identification. Then there is some aspects around the composition of that product. It also can be impurities, manufacturers, organizations involved. There is a lot of details and subcategories around the individual batches that are made. We might be then going into what is the process. So essentially, there is a batch formula, there is a manufacturing process, and then there is one instance for one particular batch that is made. But still, these are in a way little separate data blocks. And also then a few other sections of the CTD, which then lead into specifications of the product, stability study, the container closure systems used. So essentially the whole P1 to P8 or S1 to S7 section can be spread out in this way. It's a little bit different layout to look at all the information. And each of these will have its own subcomponents associated, which we could refer as a subcontent block. And then further down, based on individual sections, how granular it will get. It depends. If certain sections are big, like say stability protocol, then there will be few more data components associated with it. But essentially, we can call it as a Lego block, individual components being individual Lego pieces, but they can be built as a modular blocks, and then everything comes together in the big picture to provide the information on this drug product. Now, coming back to, sorry. So coming back to what is the focus for the PQCMC, the PQ data set that we would go through in this testing event. It's again on the manufacturer related information, specifications, batch analysis related information, and definitely the stability data summary, as well as the detailed data sections. And again, to walk through, we will start with the stability sections and then gradually trace backwards as the other data pieces come into picture. So I'll just share I will reuse the Word documents that were shared as the synthetic data. And just to walk through how we are looking at it, and then that will help us to kind of see how the XML codes or the overall hierarchy is written in the actual FHIR standards. So again, currently we use the bucket of CTD sections calling, okay, it's a S71, that means it's a stability summary and conclusion for drug substance. So that's again becomes one kind of a data block to think about, because this linkage is needed when we are talking about any of the individual data components, that what are we talking about. So it's particular drug substance, the name would be here. And then we are talking about the stability summary and conclusions. Then typically whenever the first high level summary of stability study for a drug substance would include, first is how much data is available or presented in the submission. There will be some information around number of batches. So overall, like what's the data set is made up of. Then again, depending on batches or the development history, there could be different scales the API may be made up of. So here we are saying that, okay, this is all at production scale, but there could be that, okay, 2 batches at development scale, 1 batch at production scale, and those type of details are typically captured. Then second, like associated part to it would be synthetic process. So here it's saying, okay, it's a commercial synthetic process. But again, if we are talking about, say, historic development and then the full package on stability, it could be development batch manufactured with process A, then production batch made with synthetic process that is proposed commercial and such. So all these type of details here currently are drafted as a narrative. But again, referring back to that previous picture I was showing, this is in a way linked. So although it's like it appears to be separate, in the back at least it has that connection. Then of course the next is if there are multiple sites, we would see some variation on where the API is manufactured. So again, it could be a production scale made with the same process, but at 2 different sites. And hence we need to start defining each of these data pieces to make it unique enough. The next is, so this talks about all like the actual material information. So this sentence is packed with the information about the packaging system. Again, for drug substance, usually it could be the bulk package, but still we want to refer to whether it's a proposed commercial, is it again interim package or multiple packaging system? So all these details could be considered around the container closure related details. So that data block. The next, usually it goes into the results of what type of studies. So here we are saying long-term and accelerated studies. Again, these could be like a couple other, like there could be some photo stability, there could be stress, force date. So we would see some variation as different sponsor companies write these documents. But again, all the stability studies going back into the ICH guidelines and such, we could leverage their terminologies and bring some standardization. But still this talks about like what are those types of studies, again at a very high level, not the operational aspects of studies. And then what type of stability? Is it only chemical? Is it photo? Is it physical? So these like additional details related to that stability study. And again, we have the data presented. Here it could be very specific that what is the data, the data for particular storage condition, particular packaging. If we have different batches starting study starting at different times and such, some variation in that. But again, so the storage condition, packaging, batch information, and the available data, it kind of is brought together to define what stability data set we have presented. Some conclusions, again, all results complied with the specification. So, I mean, it's a single sentence, but it then links completely to the specifications of the drug substance, hence a complete different data block. Now, based on all these summary results, there is usually a conclusion or a binding information, if we will. So this then can be, this is used to define retest and period and storage condition, and then we would put some information. This ties into the expiry dating, and hence again ties into that individual batch information, that this is the like a standard 36 months, but based on the manufacturing batch of date of each material, the actual expiry for that or actual retest period for that batch will be calculated. And again, a very specific confirmed storage condition. Here we might have multiple conditions tested, but what's the storage condition that's proposed? Now, this is still a summary section. If we go into the associated detailed section on the data, again, referring to the current CTD of S73, and I just just marking couple of points in a different color. I was trying to just highlight that some of it is new information when we are talking about this section particularly, but some is already, we can say, oh, I reused from somewhere else. So the goal is like, it's not all the time completely new information, but we are almost copying, pasting from some other documents to make this document complete. Again, the references are usually to the manufacturing process, packaging information. But what is unique now in the stability, actual operational details and such is, now we have to link with, say, the protocol numbers. So these are operational aspects of the stability study. So if there are multiple, they can be different numbers, individual batches involved in it. There can be, again, the one protocol might have 3 batches. There can be another protocol with only 2 batches and such. So again, the protocol number ties to that study design. Whenever we talk about the batch, again, some of the information that was appearing in the summary section on a high level, but is usually also listed in details, which is then the actual sizes. It can be the scale, as I mentioned, either production scale, development scale, sites, multiple sites can be might be involved. And these are the unique instances because now we are talking about the specific batch. It's not the general time frame. So, okay, when the batch was manufactured, and to tie with this protocol, again, it can be the same schedule of testing, but this study may have started early, this study may have started later. So again, when the actual that study started on this batch. So that is, we can say it's a little bit of a operational stability study, operational aspect. And then tie into the summary, where we saw that 24 months of data is available. But here also it's within that data sets, we could have some information around these lines. Now further down is now talking about the actual protocols. So again, protocol means it's the schedule and at what conditions. So of course, when we specified the batches, we also specified the packaging. And now the type of study, is it like how ICH defines respective conditions, storage condition details, and this is telling the schedule, and this is telling what is tested, because every time every test may not be conducted. When we say this, again, it's packed with all the information and many of the subcomponents because it all relates to the stability specifications. So here is just footnoted that this is our details, but all these tests usually coming, are linked to the drug product and drug substance specifications. And hence that data block is very relevant. Further down, and again, I mean, it looks simple. Maybe one point to note, right? Just putting the parenthesis for optional testing. It's very simple here, but it needs to be kind of treated when we are developing the IG or the hierarchy or model. We have to now have the, say, almost a parameter, yes no type of parameter, that is it like a must test or is it optional. So all these, like every single piece of information that is currently presented in the word or a narrative fashion, we try to bring it into the codes you saw and the hierarchy or model you are seeing in the FHIR IG. And these are the things we will also look into today. Now, another maybe just a point, usually after the, in the data section, there is also that conclusion, a little bit of narrative paragraphs usually, but as I mentioned here, this is almost like a reuse of information. So we can say it was written here and then copied into that original summary section, or maybe it was written in the summary section and again copied in this section also. So it's just that manual copying, pasting, and trying to maintain the consistency, which is one of the challenge in our current process for drafting and managing the submissions. Few new points, maybe just to mention now in the actual data tables. Again, as I mentioned, this is specification, the full block of spec parameters would appear. Now, this is a little more granular than it was in the earlier section. Sorry for scrolling up and down, but here it would just say listing the tests. Now we are going into, okay, what is the acceptance criteria? Now, when we are talking about acceptance criteria, that means, again, if it's region specific, we already have to have that link established. That what's for US, maybe what's for commercial, clinical applications and such. So all that has to be figured out so that we know that this is the correct number if we want to create this table using the IG or the codes and not manually transcribing. Few, again, operational aspects is like here, testing ongoing, very simple to say, but that means, again, it has to have the link to the actual protocol and the study start date and all that. All those type of parameters. Protocol number is here. So essentially this plus additional details are linking to this. Mentioning these for few reasons. Again, it's a reuse of information. The other point I wanted to mention is some of these are a little bit, we can say, as a standard, like almost there are very few options here because we have a good guideline or we have good list of terminologies that can be used under this parameter. But in some other cases, see here, we would see a lot of variation. So it's almost free text, hard to standardize. Okay, so these will be just the table repeating. I'll switch to drug product now. Again, now the purpose of this also bringing this document. So I was thinking that one thing is worth mentioning is, although it's now the product stability, the previous document we saw was the the substance stability, but still we see that the backbone structure of these documents or the data sets are very similar. Meaning that, okay, when we are talking about stability summary, there will be some information around the duration of, like, how much data available, batches. Earlier we had the API, the DS batches. Here they will be product batches. So that means we will be adding the details around the formulation. It can be tablet, it can be capsules. If they are multiple, we would have like all those mentioned here. There will be the details around the strength. But if you see here again, the production, the type of manufacturing, the process, the site, all those are parallel components. So if we know how to manage, say, DS and DP, like DS manufacturing information in the, like if we can automate that for drug substance part, we could use the same logic when we are presenting the drug product stability data. And that's how the current layout is also built. Like a similar logic when the same or same data components are, we kept those as like almost repeating blocks with, of course, these additional details added. Same thing, packaging information, type of stability. Here, there can be a little more discussion on the data because now it has more variations, multiple packaging and such. So sometimes there can be a reference to statistical analysis. So again, this can be considered as like a short narrative text, almost like additional comments that are added based, in addition to the actual data plots and charts if companies are sharing. And again, it will have its own shelf life, storage condition, and this information then we can say that it links to also the labeling section, which is beyond CMC. It's a non-CMC section of the dossier, but the conditions could come from here because this is the supporting data for that module. And bringing up the drug product stability, again, with the same logic, we see repetition of, say, protocol, batch information, packaging, and such. But in the drug substance, sometimes we refer more on the process, like manufacturing, synthetic process, or those type of details. Here, there could be multiple compositions, so it links to the actual composition, or there could be a formulation identifier and such. So again, this is a different data block. One thing is like, so although DS and DP both are like running parallel, what's the connector is really the drug substance, say, batch number. So anytime we are talking about the product, the linkage to the previous information is through just one point, the DS lot number. So what's here is now helping us to go back, or rather requiring us to go back to this section where we had all the data on that particular batch of drug substance. And again, this is manual currently because we have to check when we are writing these documents at, say, different times. So essentially that linkage has to be through reviews, confirmation, QC checks, and maybe managing some Excel sheets offline until we get this information, that we might have product information. What was the batch used to make this product? And then we search for the DS information on that. Again, here the focus of information will be now more on the drug product, but just by referring to this, we link to the DS information. So all this could be when the actual drug product is made, but we would also know, if needed, that relevant information on the drug substance. I think this is fine. Again, I mean, it's a similar logic and similar information as drug substance. Protocol, same thing. The test, as I mentioned, this is again going through the specs.
Hey, Sheetal. This is Rita. A very quick question from the chat. A couple questions about the color coding. Could you talk about are the colors signifying anything?
So, and sorry, I wasn't monitoring the chat. So color coding, earlier I was really, thinking of having color coding to really define everything properly. But after a point, just wanted to keep it high level. So how here I started is, okay, some of the information that was in, say, yellow could be new information on that, like in this view, but some of the information is either already reused from somewhere. So here, this is coming from another block, or it is leading to another, like a content block. But I mean, maybe, yeah, one thing is not to read too much into it for now, because it's more like the goal was to more highlight and just show the difference. It's not explicit, or it's not representing the model or the mapping. And here, say with the green, I just wanted to differentiate that from here. So this is where I mean, we thought, I mean, I wanted to bring up our discussions that this is really a little bit defined better with ICH guidelines, or less variations. This is where wanted to share that the link to the specifications, but and then it's almost like a different data block. So hence I chose not to just color it with one color, but I would say take it as a high level, and not saying that if this is green and on the other page also it's green, it's one and the same block. It may not be defined or refined to that level. I hope that helps.
Thank you.
Any other question, Rita?
I don't think so.
Okay, I think one more point is sometimes, again, depending on the complexity, that in the, say, the API example, we saw that everything can be just listed as all the results passed, and these are our data tables. But sometimes we see that individual tests which are conducted on the sample, but there could be additional discussion which is unique to that test. And hence, this gets separated, and there is some narrative, there could be some trending analysis and such associated with the tests. And of course, all these tests then lead us into the analytical section, which will be not just the specification. So specification is for sure where we saw as this list, but in addition, it will lead into the test parameters, the testing sites. So it's up to us, like how much to integrate and take it back in terms of automation. And I guess one more point is here. So now this is looking at, say, one particular method and multiple samples tested. And so again, it's a different view of looking at the data. But as notice, see, we see a lot of the information around the batch numbers. We see connection to the storage condition. We see connection to the test method. So again, it's just bringing different data components and sub blocks, or even the bigger blocks, together based on the needs of the purpose of that document. So here, because we want to look everything what happened on for assay on all these batches, that's the snapshot. Maybe sometimes we want to look at only, say, what happened to this batch. So we don't care only about the assay, but it's multiple parameters that are tested. And I think having these parsed out into the subcomponents will just help this thing being less manual and more automated. And I think that's it. Oh, one more. I think same as drug substance. So again, linking to the assay content, acceptance criteria, then leading really to the specs and all the other information, which is I mentioned also in the DS part. So I think those are the points I had.
All right. Thanks so much, Sheetal.
All right.
I think at this point we can transition over to Rick Smithies.
I'll stop sharing.
Okay, sounds good. And then Rick is going to go through the HTML renderings.
Yeah, thanks, Rita. Let's do that then. Can I share? That. Okay. You should be seeing a first slide. Does that look good?
Yep, we can see it.
Okay. All right. Let's get into it. So just a few slides, and then I'm going to go through the data sort of, for real. Okay, moving on. Yeah. So where is this data that I'm going to be showing you? It's been shared in the box folder. I won't sort of reshare that, but you should be able to find that. It looks something like this on the screen if you navigate through a few folders. And Sheetal's been taking us through this sort of some of the content in this Word document and explaining the kind of really the regulatory background, and that's all very good. I'm going to be taking you through, well, primarily this HTML folder, but the HTML and the XML are sort of intimately tied together. They're really two sides of the same coin. So hopefully you can find that. I won't always be looking at it on Box. I've got these copied onto my local PC, and you can copy them onto your local PC, one by one, or download them as a block, I expect. And then later on, they'll be on the FHIR server. Some of the same data will be there, and perhaps you'll be, when we share that later, perhaps you'll be doing the same thing over the next day or few days. So that's where it lives amongst. I mean, you might have had it emailed to you if you've had difficulties with Box, but hopefully you've all got access to that by now. So the three folders that we've seen. So yeah, we've just had our tour through a stability report, giving us this kind of like regulatory background and what all that kind of stuff is, and some of that will have been familiar to you. So, and that's kind of very much the sort of the now in my mind. There's been a couple of questions in the chat about how do we get from the kind of the here and now and the Word documents into the kind of structured universe. So I'll perhaps say a couple of things about how I see that going. But yeah, we're exchanging Word documents now, sort of maybe wrapped up in a folder, maybe sent to some server, but it's essentially a bunch of, Word or PDF documents. And part of the aim of this project and, the FHIR data in this project is to show how the same content can be exchanged more effectively, more usefully. So when it arrives, like the little graph we just saw, you could send a graph, but if you send the data points to the graph instead of the graph, or as well as the graph, then somebody else could turn that into a different graph, or alter the axes or something. So hopefully it's generally understood how structured, discrete, individual data items are more useful than some sort of pre-rendered, human written text. So Word is our kind of source. We want to be able to, we can't throw away any of the existing sort of, communicated information. We just want to communicate it in a more structured way, but also with the same level of sort of presentation. So it needs to be human readable as well as machine readable. So the plan is to move more towards an XML based exchange. And we realize that there'll be there'll still be text in there. People will still want to have textual conclusions and all the rest of it, but interspersed with individual dates, times, numbers, batch numbers, and things like that. So I'm going to be showing you a little bit about the XML, the FHIR XML, and these XML examples, they're pretty long. There's no doubt about it. I mean, sometimes they're long because they have a lot of data in them. If we're doing multiple tests over 3 months, 6 months, 24 months, then you naturally have a lot of stuff. But it's quite a verbose thing. It's mainly XML is obviously mainly meant for computers. So a point I'll make a few times perhaps is that what I'll be showing you isn't what your end users will ever see. This is meant to be sort of for the machines, and there'd be some sort of user interface over the top. So we won't be editing XML ever. We're doing that in this preparatory stage, but we're laying the foundations for the XML to be sort of hidden behind the scenes. So the XML examples, they're kind of long. I mean, I created them mostly by hand, and they're meant to match the same information in the Word documents, more or less, given the parameters of the examples, and we haven't repeated everything multiple times and so on. But they're meant to be the same sort of information as in the Word. And then the HTML pages I'm going to show are essentially a pretty printed version of the XML. So they're the same data produced by a little off-the-shelf FHIR product and a site that you could go to and use it yourself. It's not specific to this project. It just takes FHIR and it turns it into a series of sort of, schematic, color-coded kind of line-by-line things. So the HTML is mostly just a view of the XML to make the XML easy to read. It's not meant to be an end user presentation for how this would really work when it's transmitted. It's more about viewing our data standard. So perhaps this speaks to what we were saying a minute ago about how we get there. You know, why are we even doing this? What's the point of this data? So we're creating a data exchange standard. So a set of FHIR profiles, that's what they are. It's like a recipe for exchanging structured data. So we want to move to more structured data. I mean, it won't be wholly structured, I imagine, at first, you know, but just a few bits of discrete data mixed in with, you know, the more textual perhaps at first, and moving on to a fully structured one day down the road. We want to send this data losslessly, and we don't want people to be rekeying as well. You know, we don't want people copying out one system, annotating a Word document, sending it, and then when the Word document has to be resent later, they have to type in stuff. So, you know, we want to capture data in a more structured way, store it in a more structured way, and exchange it in a more structured way. And just because we're trying to exchange it or coming up with an exchange standard for structured data doesn't mean our data in the back end is going to be instantly more structured, but the two have to go together. You know, there's no point in building up our level of structured data and then you have to kind of flatten it down to a PDF and lose all the details to send it. So we've got to have this structured exchange for when we're ready to send structured data, and hopefully the two will kind of like, you know, bootstrap each other. So yeah, we don't have to structure all the data. You know, I'm showing you mostly completely structured FHIR data, but you'll be able to miss bits out, and you know, only maybe you have dates, but maybe you don't have numeric values, or vice versa. And we haven't quite come up with, you know, the level of structure that we plan to use, so we're coming up with more of a toolkit that supports different levels. Also, the data is not a program; it's not an application. It's just data. It's not meant to be, you know, this is how you would build your software. It's the data that some software would use behind the scenes, and it's just for the exchange. We're not really anticipating the data being stored in this XML or even in this HTML and then edited and put backwards and forwards. The idea is that you would have the data in your systems, existing systems or new extensions to those systems, prepare the data in there, and then at the last minute you would turn it into XML, send it, and the XML is kind of disposable. You know, you'd send the XML, somebody receives the XML, they take the data out of the XML and put it into their system, and it'd be those systems that are holding the data. You know, we're not anticipating a bunch of XML files as the storage of the data. Yeah, but this last point, you know, we're coming up with an exchange model, but because we're having to come up with all the concepts in a, you know, in a formal way and decide which goes where in the data structures, it also makes a reasonable sort of, you know, model for this, a structured model for this domain anyway, even if you're thinking of, you know, not just exchange, but maybe the storage and so on. That's one of the things about FHIR, it's meant for exchange, but once you've come up with a standard model for exchange of data for that domain, it also makes quite a good model for that domain in general, not even talking about the exchange of data, storage of data. Okay, so some sort of high level points, and those are sort of my opinions. What does this HTML I'm going to show you show? Well, it's the HTML sort of here on the left, if you can see my cursor, and it's the XML behind the scenes. So we've got, these are the two folders, the HTML folder on the left and the XML, or one of the files in the XML folder on the right, but it's the same information. So you can sort of see, you know, in this view here, the kind of more human readable, you've got an organization thing here and an identifier thing here. If you look at the XML, and you have to sort of find the right place, but you can see, well, that's called organization because this is called organization. And the identifier, it's this number, and you can see it over here, identifier, and it's this number. So some of the details are hidden just to kind of make it more readable. But the reason it's identifier name contact is because in the XML it's identifier name contact. So there's a more or less a one-to-one correspondence between what you see in the HTML and the XML. It's not too far behind. It's not too different. And we've done things like collapse the address down from 5 lines into one long line just to kind of, you know, make it all fit on one screen. And then we have another organization later on in the same file, and that's why you've got another section here. So there's a real correspondence between the two. And I'll show you this when we get there, but if you were to hover your mouse over at one of these fields in the HTML, it will pop up a little mini view of the XML. So that helps you to orientate in the XML, you know, where is this address coming from? Is it this address or whatever it might be? So you could have the two files open side by side, you know, your HTML on one view and your organization XML on the other view, if you like. That would be good, but the little pop-ups guide you to which bits are which. Okay. Yeah, so what is this HTML? Well, as I say, it's an HTML view of the same stuff. So it's not a different thing. It's just the same stuff presented differently, which helps, you know, it just helps read it, but also it helps with things like links, because in the XML, you know, you might see a reference in the XML which corresponds to something 3 screens below, which you're just paging up and down and searching, and it can be quite hard to follow. In the HTML, those links are sort of pulled together. So a reference to a batch, it would probably pull the batch number through and display it in that bit rather than lower down. So it just helps kind of visualize it. And yeah, it's meant for exploring the data and showing you the XML data. As I say, it's not meant to be what real users would see. That's not the point of this. I mean, it makes a reasonable, nice view, but that isn't the aim of it. And as I say, people should not be editing XML or even this HTML. But we're kind of data aware people, so it shouldn't be too scary for us to get down into the XML and get hands on. So we're showing the data standards, the proposed data standards. And yeah, it's not how it would look in real life, but that's okay. There are actually a couple of sides to the HTML. Some of it's generic, and some of it's specific to this to these particular reports and FHIR data for this project. So you might see this, you might recognize this from the Word document that Sheetal just showed. This is a sort of secondary view over the ordinary HTML view, which I'm going to show you. This is an extract from something you'll see again in a moment. So the XML in the background has an action with this title, followed by another action with this title, followed by another action with this title, and even a fourth level here. And you can see this is kind of makes sense. It's a test, and it's got a long-term test, and at the beginning of the test you do X, Y, and after 3 months you do X. And that is just about readable, but it doesn't really show the picture. And well, as I was preparing this, I was thinking, well, would anybody be able to, reverse engineer this and come up with the data, or does it even hold together? So we added these extra sort of displays, and the thing you see here is generated from this thing you see here. It's not just been typed or cut and paste in. There's a little bit of code that just pulls off every third one and puts it in this column, and every sixth one and puts it in this column. So when you see these kind of slightly more colorful tables, they are just another view of the same more raw data. So this is kind of our test. So I could see this and it looked all right, but you never really know whether a receiving system would be able to correlate it properly, or whether you've put something in the wrong column. So that was kind of the point of that. So you'll see that for real in just a moment. But yeah, about the FHIR, about the XML, before we get into the actual data. FHIR is a bunch of these resources, and we have quite a lot of them in these examples. We have a few, I don't know, maybe a dozen types of resources, something like that, and lots of instances of them, maybe 20 or 30 in one of our files. So a bit chunky, a bit big. And they live in this thing called a FHIR bundle, which is the kind of the top of the tree and contains all the other things. And each of these resources, and they correspond to a colored chunk on the screen. So when we see our colored gray or yellow blobs, there is some, the color coding doesn't really mean anything, harking back to what we were getting questions on before, but it just, when you see the same object, it'll always be the same color. So if you see one of these things, a medicinal product definition, that'll always be yellow. And I can't remember what these colors are. Maybe that's green. No, that one's green. They'll always be green. So you can see where one stops and the next one starts, and they have some consistency, even if the colors are somewhat arbitrary. So these are FHIR resources, off-the-shelf FHIR resources that we've been, some of the members of this project have been helping prepare these resources to get them ready for this project and other projects, but they're suitable for use in many different things. So sometimes when we use a resource in this project, it might not have the name we would expect. We've not invented these resources especially for this project. And that's not the point of FHIR. FHIR is a general collection of things that you can use on for any sort of medical informatics thing. So we have plans, we have observations, even though we might call them specifications or tests or some other word in our domain, but necessarily because we're using off-the-shelf generic components. That's why things don't always, they're not always the same names that we would call them if we were doing them from scratch. But then we get the reuse. The bonus is we've got this stack of FHIR stuff that's out there. There's lots of tools for it, lots of free stuff, lots of source code. Other industries are getting into this as well. Other sort of government supported projects around sort of healthcare and general patient stuff all use the same thing. So there's an industry shift, and it's going towards FHIR, and it's good if we can sort of jump on that. Yeah, so these are, if you want to know which resources are being used as a sort of overview, then this link here to the IG, the implementation guide, lists them and shows the properties of them and so on. So that's a good place for an overview. And then in the HTML views, they're the actual resources are, so in different colors, and they start with the bold names. So if I just go back one slide. Yeah. The reason that's in bold on this organization thing here is because it's the name of the top of this bit here. It's the resource name, and the other things not in bold are the sort of properties within it. So the resources are your bold things. So a sort of high level view, I mean, there's quite a few resources, as I said, maybe a dozen or so, I can't remember exactly. But we've got some categories, high level areas. We've got, the definition of the product. So what the ingredients are, its form and all the rest of it. The substance, similarly, what chemical composition is and so on. And they're obviously somewhat related. So, but a couple of categories there, and then perhaps a couple more categories. One where we're getting into defining tests, what we're going to do. So we're going to do this test at 3 months, and the answer must be less than 0.1 something. And then at 6 months, we're going to do some other test, and at that time it must be less than 0.2 or so on. So we've got specifications, definitions. So there's a group of resources around that. And then we've kind of got the counterpoint to that is the actual tests that you do, and almost like the answers to these questions. So this says what you do at a certain time, or it's like a template for what you do, the protocol, and then you'll get the actual observations that say, well, it really was this value and it really was this batch, and you'll group that together with a conclusion into some sort of report. And then we have some other sort of sundry things for organizations and sort of stuff that pulls it all together. So within that, then, if you want to know about the product and substance FHIR resources, have a look at FHIR. I mean, all of this stuff's in FHIR, but in particular, if you go to FHIR, this is your link. On the front page, there's a link to this thing called medication definition module. That's not the module, but that's the link to the module on the front page. You'll see if you scroll down, and you click on this link here, and it takes you to a page that explains something about how these resources fit together, how we do packages and things like closures, how we do the tablet, how we do the ingredients for the tablet, how does the ingredient relate to the substance it's made of. So this diagram is taken from that page, so you can sort of get an idea of products with packages and tablets in them and so on. We won't teach you that today, but it's a good resource. And then I did do a slide especially for this about the plans and the observations, because that's the data I'm going to be showing you has a lot of this stuff in it. So this quite complicated, but hopefully understandable. We've got some drug, and we come up with a stability study plan for that drug. And plan means sort of the things that you're going to do or you're expected to do at certain times. So there's a resource in FHIR called plan definition, which we'll be seeing a lot of. And then it has a series of actions in it. So you can say, well, do this then, do this, repeat this one, do this every 6 months and so on. And each action can be an observation definition. Observation is, doesn't mean sort of looking at it, but it means testing it, seeing what its pH is or whatever it might be. And, this observation will have a test and a code to say what the test is. All this would be coded ideally. And then some criteria. Is it meant to be observed as being an orange solid, or is it meant to be observed as being, pH 6.9 or something? So this is kind of like the definition of what you should be doing. And then over here, we have sort of real batches of this, somewhat abstract definition of a product. This is a product. This is the actual bit of the product you could touch, and put on a spoon or something, or you might have a kilogram of tablets, and then we run actual tests on those, on batch 1 and on batch 2. And what you do is defined by these ones over here. So this is the template in the middle for this test on the right, and then the actual numbers come back from the lab. And you probably do more than 2. You'd probably do 22, and then you wrap those in a diagnostic report resource, another resource, together with some text. We have found that the test came back fine, and we didn't do this test for this reason, but that's okay. And we've come to the conclusion that the shelf life it's fine to be 36 months because the numbers back that up. So that's a lot of the stuff we're going to be seeing in HTML has that kind of basic form. The stability study, for instance, is and we saw that in the Word document. We saw a kind of plan for what you're supposed to do when, and then you get the actual, what actually happened, or what's still in progress. Maybe we haven't finished the observations yet. Okay, well, with that said, let's go and look at some real data. Let me go into my folder. So let's start off simple. So I'm getting this from the files that are in, here's my machine. Hopefully that's still showing. And you can see I've downloaded the box stuff, or and I've got this one. Let's take you through a simple one first. Put that back over there. Put that back over there. So very, very simple. This is an organization. Perhaps I'll also show the XML at the same time. Well, there's going to be a lot of windows around, and I'll be flicking things backwards and forwards, so you'll have to bear with me at times. So here we go. So it might be hard to show these at the same time, but this is the XML for that. So let's briefly look at the XML. You can see it's a FHIR bundle. Hopefully that is the size that can be seen. And there's going to be a lot of sort of nuts and bolts in here that might not mean that much to you, but we're more about the principles than the details, perhaps. So we see bundles of things. Bundles are our FHIR groups of other things. And then we think inside them we've got things like organization, and this has got some sort of made up thing, and it's got an identifier, perhaps one that's been assigned by the FDA, and then a name, and then an address. And we saw that on our slide a little while ago, didn't we? So that comes out in our HTML looking like this, pretty obvious. So you got, what's that, 6 or 7, 6 organizations. And you can see if you hover, you can see that, yeah, it's just what you saw a minute ago. You can see it's an organization with a certain ID, manufacturer, and then you can see that 681 Main Street, Lumberton, and it's the same as what you see. And if you hover over the actual name, you'll see just the bit that's the name. So it kind of focuses down on that bit, and similarly identifier is going to do it for me. Yeah, focuses down on just that bit. And then a repeating set of organizations. So that's our kind of basic how our HTML works. Let's move on to something more complicated. Let's try should we go straight for stability? Let's go straight for stability, which is one of the big ones. Okay, so this might look familiar because it's meant to mimic what was in the Word document that you saw quite recently. We've got we've got a resource at the top here, and we've got a sort of a high level HTML thing that's designed to exactly mimic the Word, just to kind of say, well, the data down here in the rest of it can actually be sort of summarized even further like this. And the fact that it looks pretty much like the Word document just kind of gives us an easy way to say, well, the Word document looks like this, and this looks like this, so therefore they cover the same ground. In this, and we don't miss anything out. But there's no real reason it has to be presented in this way. I just recreated the Word document to say, well, you know, that just so you can see that they look the same. There's no reason it has to be in this particular format rather than any other, because the information in here is really the information that you're seeing here. So you can see that there's 3 columns because there's 3 batches. And though the actual batches you're seeing with these, where are the batch numbers? So the batch numbers, yeah, here we go 3, 3, 4, 4, 5, 6, and 7. You know, the reason it's that is because we have a FHIR resource that has that batch number manufactured on that date, and the date you're seeing there is the date that I just also pulled into there. And again, so we'll somewhere else say my site. There we go, my site and my site and so on. So there's multiple views of the same thing here, and this behind the scenes is one of these. So there's a FHIR resource behind the scenes that's a batch of product, so you can have multiple ones of those, and then we can see, well, which product is it? You know, what is it? So it's Stelbat tablets, 20 milligram, and I think that link will work. Yeah, it takes me down the bottom or lower down the page, and I've got some information. So you can see I've jumped down a couple of screens, and my scroll bar has gone down a bit. You can see it is this product, which is a medicinal product definition resource. You can see that, and then it's got an ingredient, and that in turn is a substance, and you know, it's an active ingredient of this stelbatolol and so on. So it links together. I can expand that and go into the product, this little plus here. I can see, well, it's got a name type, and it's got a strength 20 milligrams, and it's got some description, and it's a for oral use, and it's a gastro-resistant tablet, perhaps. So some of these things you can go into in a bit more detail, even down to the substance and when was the, you know, who manufactures the substance and so on. So let's scroll back up where we were before. This is where we were. So these are the 3 batches that are the focus of our test, and you know, where they came from and so on. So let's scroll down past those 3 batches, and then in this presentation, I've got different versions. There are different versions of this data over the time. The one you're seeing on the screen is as of a couple of weeks ago. When you see it, I'm going to send it to the server shortly, or perhaps that, you know, in the next little phase of this presentation. You might see a few minor differences because these things are slightly evolving, but the principles are the same. So we have our protocol, or whatever you want to call it, our specification for how we're going to do a stability test. And you know, it's called this, and it's got some description here. And then it's that, what is the subject of this protocol? Well, it's about a certain product, and you can see the product again. It's actually, I think it's showing the product twice, or maybe that is the one we jumped to before. Perhaps, yeah, it is the same one, I think. So we've just been there. So this stability study is about that. I think I can close that up. Yeah, move it out of the way, and then we get into the kind of the bit that's quite complicated. It's, well, what are the steps? You know, how does this happen? This is our summary of it. So we've got 2 sort of phases, if you like, a long-term one where you actually keep the sample and just let time pass, and then an accelerated one where we sort of cook it a bit and pretend that 3 months has passed. I think that's how it works. So inside our resource, we have a quite a complicated structure, and you can see some notes and some comments there, and it's got a, well, when am I doing this? I'm doing this at 0 months. And okay, when am I doing this one? And I'm doing this at, and you can probably see there I'm doing this at 3 months, and we're actually doing it low and high. We're doing it from 3 months to 3 months. So that's a point in time at 3 months. In theory, you could have one action that took an extended time. We probably don't need that. So we're doing an initial one, a 3 month, a 6 month, a 9 month, a 12 months, and an 18 months. And let's dig in. And then the thing repeats, actually. So the accelerated one comes after it. And then we can look in and see, well, what are we actually doing? What are we doing there? At the initial one, the timing is 0 months. The frequency is once. You can have things that repeat. All of this action and plan stuff is generic FHIR machinery that you can use for any sort of plan. So it could be caring for a patient. It could be doing an operation, the seconds in the operation where you have to put the knife in and then 10 minutes later you do something else, or 10 minutes before you do the anesthetic and so on. So it's a generic timing plan and steps structure. So yeah, and then what do we do at this time? Well, at this time we do the following, and these are activities. So this is another resource that gets more into the details of what we do, and then that connects in turn to a third resource, which is an observation definition, and it's got this abstract name X because that's what the original plan had. This is, the original Word document has sort of mnemonic codes for things that you do at 3 months, things you do at 6 months. And this is a group of things that you do that we're just calling X. It's a battery of tests. It's more than one test. And you do it on a certain specimen. So another FHIR resource, a specimen definition. And that tells us what we can do with our specimen. So this, a specimen here is like our prepared batch, the batch we're going to test. Specimen might not be the word that you think of here, but when you take some sort of sample, whether it's of a patient or of some water from a lake or a tablet from a jar, whatever it might be, and you're going to test it, it's called a specimen in FHIR. And what are we going to do with it? Well, we can handle it. We're going to do this to it. We're going to keep it at that temperature rather than some other temperature. And then, well, what tests are we actually going to do? We're going to do the following tests. So we're going to do a description test. So that's not really much of a test, but you're going to examine it, get your magnifying glass on it, and check that it really is a tablet. It's not degraded. It still looks like a tablet. It's still got 175 written on it. And then we're going to check for impurities with another test, and we're going to have different impurities with different allowable impurity levels. And then we're going to check the water content. So that's our X test. So we've made a little repeatable X unit, and we've got a Y unit down here, which consists of some other things. So we're going to check the microbiological things and check that it complies with the European Pharmacopoeia and some other things at Z. We're going to, what we're going to do here, we're going to just check that uniformity of dosage, whatever that means exactly, I'm not totally sure. And these are sort of abbreviated from what the full protocol might be, but there's only a need to sort of illustrate it really. So yeah, so in the end we lay out a series of steps that mimic this structure here, with like doing those 3 in the first 0 months, doing just the X bit at 3, at 6, at 9, and then the X and the Y at 12 and 18, but we don't bother with it on the accelerated at 12 and 18 because that's just the protocol we happen to have chosen. So those lay out in a machine readable way what we're going to do to this, for this test. And you can imagine building that up with some sort of user interface that said, oh, well, I'm going to have an X at this month, I'm going to have a Y at this month. And you could cut and paste it and vary it from one substance to the next and say, well, for that substance, we're going to do this, and I'll save that in my database. And, for the other one, I'm going to do something else. So you can build up a little library of these and build up your X and Y's, pardon me, from your different tests, and pick these out of some sort of test library. So you'd have perhaps LOINC codes or some specific codes to say, well, we're doing water content, we're doing pH content, we're doing whatever it might be, standard codes. So that goes down and down, and you build up a similar kind of thing. And then we say, well, who's going to do this? And it's this testing site here and something there. And again, if you hover over that, you'll see it's an organization, as we've already seen. And then, so, I mean, this report is a report. It's of a test that happened. So we've got the protocol. The plan is in the test, but you don't necessarily have to have the plan in the test. You could just refer to the plan somewhere else and say, well, I sent you the plan. I logged the plan 6 months ago, sent that to you 6 months ago, and I'm just going to refer to the plan now and say, here's the plan definition number. We've chosen to embed it in this actual XML here, so you can see the plan followed by the results, but that's not perhaps necessary. There's a bit of redundancy in there, perhaps. But here's the answers. Here's the result of the thing. So the whole response is a diagnostic report FHIR resource, and that's the same FHIR resource you do if you were you know, doing an X-ray on a patient and then writing something about it. You know, we use the same machinery for lots of drugs as we do for, you know, if a patient has something done to them and they get some numbers back. So the conclusion, you have a textual conclusion, and I've got like a, you know, a few words here that could extend to be a whole, you know, side of data with all sorts of stuff in it. You know, and you can have HTML in there if you want, and a bit of structure and bullet points and so on if you like. But that's old school stuff. You know, we can already do that. That's not really what we're mainly talking about. This is more what we're talking about. We have, well, this is our sort of, you know, really specific to this document format, where we take the results, which are a bit lower down on this. I'm just going to scroll down to them in the moment, and we match them back to our, like, why we're doing them. So we'll say, well, on the 0 month, we said what we were going to do, and we're now saying we've actually done one for this batch 33445, and for this impurity we actually got 0.1, and that's okay because it was supposed to be less than 0.8, and some other values and so on. And obviously these are not all filled in, perhaps just because, well, realistically, because I just didn't put in, you know, all the data in this example, just to give you a kind of a starting point. But perhaps this might be completely filled in a realistic one. And again, this table here is derived from, that's the first batch, second batch, third batch, from this data down below. So it's a kind of live presentation of that, just to show that you can actually turn these green things, which are a bit harder to read, into something that looks more like the old world. And again, you wouldn't have to send that. The idea is that the software that receives this would be able to extract it and build that table. You know, we don't have to send the table if we're sending the raw material for the table. And if instead of that you wanted it in different orientation, or, you know, different headings, or as a graph, you could do that. You know, this is not the presentation. This is just one possible presentation. But the actual results are more FHIR resources. So here's an observation, as opposed to an observation definition that we saw before about the ideal values that you're supposed to get. This has the actual values that we did get. And it says, well, we're doing it on this actual specimen, and we did keep this specimen for 0 months. We collected it here, and we didn't keep it at all. We just acted on it straight away because it's the initial phase. Which subject is it? And if I click back, it would take me back there. It links back to the definition of the reason for doing it. So that's one of the advantages of FHIR. You can link everything to everything. So this observation, it's about that, and I could click and go there, and it's about this one, and I actually will click and see if that works. If I expanded it above, yeah, it'll jump back to it and say, right, it's okay. The reason we're doing it is because of this. It's a long-term test, and it's whatever it is. It's the 0 month one. So let's go back to where it was. Bit of scrolling. So you can see, so each observation can be tied back to the reason for doing it. And that, as I say, that activity definition is actually in this block of XML, but it need not necessarily be. This pointer here could be linked to the standard protocol server at some company. So you publish your protocols on one server, and then your actual reports just refer to them without having to repeat it all, possibly. And then what actually happened? Well, we did the description, and we're just saying it complies. And we did the impurity checks, and we found these levels, which are all, and it goes on and on and on. There's obviously lots of it, water content, etc. And then we did it again at, I think this might be the second batch, so the same, but for a second batch, 446, and so on. And it goes down and down and gets a little bit repetitive, obviously. Third batch, 3347, same definition. Same protocol, different batch. And we got different results for this one. And then I think this one, yeah, this is the 18-month test. We skipped over the 3 and 6 months. And at 18 months, we did this to specimen. We stored it for 18 months at this temperature and humidity. And at that time, something else happened. And by this time, it no longer complied with this test, or whatever happened. And then down and down and down. So you can see more and more and more. And it's just more of the same, really, and that's it. So those are your sort of, you know, the results of the test. And as I say, you could present that in any way you like. You could turn that back into a table. You could turn it into a PDF when it arrives. I'm not saying transmit it as a PDF because that kind of defeats the point. Transmit it as discrete data to the level that you can do. If you can just send a couple of results and a bunch of text, that would still be useful. And this is kind of like perhaps the somewhat, medium term or far term possibly, where we can send complete granular data, and the receiver would be able to, turn it into tables, graphs, pie chart, whatever you want to do with it. So how are we do for time? That's we've got a bit more, haven't we? Shall I look at the next? Sort of a slightly pause for questions. I'm trying to look at the chat, but let's look at one of our other. But that was due. Are we just trying to check my time? Am I good for time, Rita, or are we moving on to the next part now? Well, the next part's me anyway, actually. So.
Yeah, I think we're just a couple minutes over, but
Okay.
I think we can take the time. I think we should have some extra time later today. So I think if we need to take a little longer here, I don't think it's a big deal.
Okay. Yeah, I'm just looking at my time. Yes, that's right. I'm a little bit over. So, but yeah, before we move on to our next thing, which again is more kind of more of the same, really. There's the other HTML. We've got stability, which you've just seen. We've got specification. Let's load that up. And the reason I start with stability, perhaps, well, it's kind of almost like the superset or the bigger one. If you can do a stability and have all the different steps in the plan and the results, then the specification looks pretty similar, but if anything, it's simpler. So we can say, well, for this product, this is a specification thing, and again, it's one of our plans. Then you can do a certain series of tests. These are our tests, and this one is just the specification. It doesn't have the results. So this is just the plan that you want to do, and you say, well, we're saying that at release time it should have this value, and at shelf life, end of shelf life time, it has this value, and so on. So this is a similar plan as we just saw in stability, and you can drill into it and see, well, at this time we're going to say it has to be an orange tablet, and here it has to be a certain pH or whatever that is, not pH, but yeah, European Pharmacopoeia value. So that's a pretty similar. That's the batch analysis. That's the specification. We just sort of showed the stability before that. The batch analysis sort of ties in with the specification because it's testing a given batch to see that it matches that specification. So let's look at that. And here's our table again. So this one exactly repeats. If I flick back a tab, you can see that's basically the same as that. Our batch analysis follows the plan of the specification. So this is just repeated again. And then you would get an actual report. Again, that's our diagnostic report, which you might remember. And this time it's a batch analysis report. Why are we doing this thing? Well, we're doing it because we have a batch analysis plan. We have a specification. And what did we find? Well, on this lot number, this happened and that did not comply, and so on. So it's looking very similar to what we've already seen, and it goes down and down and down. You can see, scrolling too fast for you to read, but you can see the idea. And then the same again for, where's my window? The same again for substance, very similar. We try to keep them sort of almost mirror images of each other. So substances are kind of different from products, but the testing of them follows the same sort of pattern. You have certain things. This is the specification for one. Certain criteria you have to meet, and it's not about products. It's about a substance that goes into a product. And again, the same sort of definition of tests. What you do at a certain time, identification, but this time by spectroscopy. And then let's go into our stability for substance, and with a similar kind of thing, you've got certain batches of substance called Cat 1, Cat 2, and Cat 3, and whether they're production or how big they are, quite big, 120 kilograms. And then we can say, well, it's this substance. Here is our batch, Cat 1 batch, and it was manufactured on this date and released on this date. I'm not sure those dates actually make sense, but anyway, you get the idea. And it's due to be retested then, and it was and so on by this company. And then we can say, well, what were we planning to do to it? We were planning to test it this way, using this slightly different but very similar plan to what you saw before. And you can see a similar thing where this is what we found for the substance, all looking very similar. So that should be familiar. And again, lots and lots of green tests with different numbers on them. And as I say, if you want to know, well, okay, well, where am I seeing that code? You know, what is that status? Where am I seeing that value? If you hover over it, you can see that you can sort of follow the tree. You'd have to have the XML in a window next to you and kind of go down and say, all right, well, it's the bundle, entry, resource, observation, containing another observation, because these are grouped together into sort of batteries. And you can see, oh yeah, well, it's that AAA code in there. You can see I'm sort of pointing at my screen and hovering at the same time. So that's a view through the HTML. Okay. So yeah, slight pause for questions while I prepare my next part about more about the data, and I'll upload some data to the server, and I can make some changes to it, show you some JSON instead of XML, and do a bit of validation. Okay, so this is due to go till, yeah, I've got another 45 minutes. Okay, so I'm going to show you this this tool here. This is a standard. It's called Oxygen. This tool. It's a standard XML tool. Not specific to this project. It's if you've got techies on your team, this is the sort of thing they might be using. Let's start simple again with our organizations data, the one I showed you before when I zoomed in, didn't it? That's why that one's big, because I zoomed in. So this is some FHIR data ready to go. It's one long bundle with multiple organizations in it. And that might be how it works. You might submit 20 or 50 organizations to somebody in one go, or maybe do individual organizations. Or I'm just updating this one. How that workflow works is a bit kind of up in the air, but we're developing tools for the different ways you could do this. Yeah, Jeffrey, yeah, Visual Studio Code is a great free XML and JSON editor. I know plenty of people use that. It's got lots of good features. So let's take this XML. I'm going to cut and paste the whole thing, and I'm going to use Postman. So we're getting quite technical now. Postman I loaded earlier because anybody who's used it knows it takes a long time to load. So this is the first time you've seen the address of our server, probably. Hopefully that's visible. So it's dxpqlantannagroup.com/FHIR. And I'm going to blank that out for now and hide that. I'm going to send this data to that server. So Postman is it's a REST tool. REST as in reading and writing HTTP data. So the FHIR server here is a web server. You can see it looks like a web address. So it's a real FHIR server, but it looks like a web address. If I was to I copied that data, so I'm just going to paste it in there to keep it. Here's my organization. But if I was to go there with a browser, let's just see if that will. Yeah, so I'll go there with a browser. I've just gone there with this browser. Let's do it again. I literally type that in there using an ordinary Chrome browser, and it comes back with a message to say the server is alive. Now it actually says bad request. I mean, that might look bad, but that's actually the correct response for this, because I've gone to the base of the server, just the FHIR point of the server, and it's saying, well, you just come to the base of the server, so that's not really right. You're supposed to be asking for some data. So although technically that's an error, it means the server is alive and running, and you could that's a good test. So a browser is a good way of reading FHIR data, because it's a browser is it's an HTTP browser. That's what browsers do. They let you get pages of HTTP data and the XML and JSON within it. But if you're going to send data to a server, as well as reading and deleting things, then you need something more sophisticated, such as Postman. And there's Visual Studio plugin, Visual Studio Code plugin, and other tools such as Insomnia and lots of different ones. But here's the one I'm a bit of an industry standard to use Postman, although it can be a little bit complicated. So you need to know where your server lives, and we're gonna. So that was the address of the server that end up to the point where it ends in FHIR. That's the address of the server as a whole. Then I've added on this bit here /bundle on the end of my server address to say because I'm gonna give it a bundle. I'm gonna send a bundle of organizations. So I need to say, okay, /bundle. And I'm creating a new bit on this server. So I'm going to post some data. It's called posting when you send some data to a server, and I just cut and paste that from my folder, and that's the same as you would find in the box folder. So there it is, like on my sort of whatever that is, 100 odd lines of XML, and I'm going to send it to that server. Now, for the more technical, I would probably need to set some headers behind the scenes. I need to tell it I'm sending it XML. I could be sending it JSON, and you can send it either, and it kind of needs to know. So you put this magic keyword in here, application FHIR plus XML, which is standard across all of FHIR, and you make sure that matches whether you're sending XML or JSON. I'm going to send XML, and I've put that in there. Check that looks right, and I'm going to hit the send button and hope that it works. Hooray! So it came back with a message here. So that's what I sent up here, and this is what's come back below, and it looks pretty similar, but if you'll notice, it's changed the ID. It's actually assigned an ID here. In fact, technically, for those of you who know FHIR, that ID there in the send, it should not really have been there, because the server generally assigns the ID if it's in charge of assigning IDs, and it will ignore any IDs that you send it. So it's assigned me an ID of 9504. And when did it do it? Well, it did it at 27th of the 4th, 2023 at 13:38, which is GMT. So it won't be your time. It's not my time either, but it's close. And it's echoed back the same data, and it's given me a 201 created. If that had been a 404 or a 412 error, it would have come back and said, sorry, that didn't make sense. You sent JSON by mistake, or you didn't format it right. That wouldn't have worked. But yeah, it's worked. So now we can go somewhere and we can look for ID 9504. Now I could do that in Postman, but just to keep it interesting, I'll do it in Chrome, and I'll go to bundle, what was it, 9504 from a clipboard, and if I browse there, and you'll see the same data. Now notice it's in JSON this time. So we sent it in XML, look, over here in XML, and we got it back in JSON, which is interesting. And you can get it back in JSON, or you can click on these tabs, or use your, I'll do it like this, XML, and I can get the same data in XML. Oh, well, it wants to save it to my disk, so I won't actually do that, but I'll use the sort of cheap way and do it in it. Yeah, I'll do it like that, and you can see the same data in XML or JSON. So you can start to see here that the server doesn't really have a preference to XML or JSON. You can send it in one, get it back in the other. It's completely transparent. If you've got some XML and you prefer it in JSON, send it to the server and then simply ask for it back. That you can just get it back in JSON. And I'm actually getting it from, you can actually fetch it from the server in that format. It's not, I'm not sure if it's just converting it in the browser or not. I think it's going back to the server and fetching it again. So here's, some JSON for organizations and how that looks in FHIR. So that's how you would upload that. Let's look at my little agenda. So say I wasn't sure whether I'd formatted it correctly, and we've got some special criteria in that we need about FHIR observations, and there's a couple of ways of checking, and this is where our FHIR profiles come in. FHIR has some rules about organizations in itself. So we can go to FHIR. Let's have a look at FHIR. If I go to FHIR here, this is FHIR, and you can look for organizations in resource types. Find it in here, and you can see scrolling organization in there, and you can see what it's supposed to be like. It's supposed to have, well, it can have an identifier, and it can have a type, and it can have it, and so on. So you can see the rules of what you're allowed to put in there and what you're not allowed to put in there. And then in our implementation guide for this project, we can make extra rules. We can say, well, the name, it has to have a name. You know, organizations don't always have a name, but when we're talking about drug manufacturing, you know, we're dealing with companies that have names. You know, these are proper sort of official organizations. And maybe we say, well, we don't care about the description, but we must have a contact. So you can make extra rules, and maybe the types of organizations that we deal with are not just any type of organization, but maybe they're, you know, manufacturers, repackagers, regulators, health authorities, whatever they might be. So we can make extra rules, but let's just see, check some basic rules on this server. So maybe I sent this to the server, or I'm going to send it to the server, but I'm not sure if it's valid. So I'll switch my other tab, and you can see I put this $validate on the end. It's the same address, but I put $validate on the end. And this is the same data I had on the other tab. I'm going to take that out because that, again, that isn't really. And I'm going to put some junk in here. I'm going to put, and that definitely isn't valid by FHIR. It's obviously just made up. And when I send that onto this $validate, this basically receives that data, checks it against the FHIR standard, and checks it against the profile as well. So we're actually labeling it here. You can see this label here. In this case, it didn't have it in the other tab. This is an extra thing. We're saying this bundle corresponds to not just the FHIR bundle of organizations, but the DXPQ definition of organizations as well, which actually at the moment doesn't vary too much, but it's kind of like where we will put our rules in when we decide that this has to be an FDA number or maybe not, and this has to have a name or maybe not. So I'll hit that and see what it says, and it will say, well, 412. So for those of you who know HTTP, that's an error code, but that's what we were hoping for. That's good. It is an error. Precondition failed. Bit of an obscure name for the thing, but it says back here, error, undefined element, junk. So not surprisingly, and it tells me it's in the organization on line 12, column 20, so it's pretty accurate in this case. Lo and behold, I put some junk in there. If I take that out, try it again, it'll come back and say 200, okay. And then it gives me some, so that's basically a success, and it's okay. It gives me some advice about my resource. It says we should have some text in there. Fair enough. These are kind of, you know, top tips for FHIR resources that don't necessarily we know about those, and we're deciding not to do that. And that's not having the word junk in there is a FHIR rule. Your FHIR does not allow you to have any old tag in there. But there is one thing that this FHIR, there's one thing that's allowed in FHIR but not allowed by our rules for this project, and that is that you can't send an empty bundle. Just to take that out, you can see me going to select all these organizations, go all the way down the bottom, down, and take that out. That empty bundle is allowed in FHIR. You can have an empty bundle in FHIR. Pretty pointless, but it's legal FHIR. It's not forbidden in FHIR. But in our case, we don't want people exchanging empty lists of organizations because that doesn't really do anything. And I think if I send that, yeah, it gives me an error, and that's from the profile. So that's from the, you know, the specific this project's stuff. It says bundle entry minimum required 1. I've got to have something in there or it doesn't like it, and it says found 0, and it says, well, it's using this set of rules here. It's using the DXPQ check, which you said you were conforming to, but you were actually not conforming to. You're lying. You said, please check me against this. It did check against that, and it said, that's no good. You're supposed to have 1, but we found 0. So we can build on that framework of things and get more specific, and perhaps in certain circumstances you have to have a plan inside your bundle, and maybe in other circumstances you have to have a substance, and if you forget the substance, that's not going to make any sense. So we make the substance mandatory and so on. So we can build on those profiles once we're, you know, get a bit more decisions about exactly what we need to send in what circumstances. I think what I might try, purely out of interest, is if I get rid of all of them apart from 1, that will succeed. I'm going a bit quick there. That should succeed. 200, okay. But if I was to change it to something else, I'll get rid of some of that because that probably. I literally haven't tried this before, so let's just see if it allows me to have a patient in an organization bundle. It might do and it might not. I'm not sure. I'm generally not sure what it will do. Okay, yeah, good. It said, that was what I was hoping. It says this element does not match any known slice. Bit of a strange word for you, slice. But it's saying this part of the message is closed and it has to have an organization. So it doesn't match the all organizations rule. That's fair enough because we tried sending it some patient data, which would never have made sense. So it's detected that you sent it some junk. So again, that's a specific thing about this, this DXPQ stuff. We don't send patients. Okay, let's. What I can also show is the little viewer program, which you've seen the HTML for. If I go to this here. Yeah, this is a little a general FHIR viewing thing. You can pick that bundle, which now then I need to figure out which ID it was. So it's this 9504. So I'm going to do a bit of cutting and pasting, put that in there, go back to that, and put that in there, and it will show me bundle 9504 with a bit of luck. Let's try that. This will take a few seconds because it does a bit of searching around. Okay, that was quite quick. So it came back with, yeah, the HTML view, similar to the ones that you've seen in your pack. But this is kind of live on the server because I only added that ID a minute ago. If I picked another ID, if I picked like 999, it would, come back and say nothing there. So it really is a live thing. Back on that, and yeah, it shows me not only the sort of pretty printed view, but down the end here you've got some sort of other stuff like that. The same HTML down below. And let's say we want to update this. Say the company moved from Main Street to some other street. How would we update that? I would probably go back to here and change that to, I don't know, maybe they just moved next door to 6, well, let's say 7. With a different postcode, I don't know, different zip code, who knows. So let's take that 700 this time, and we're going to instead of posting it, if I can find my window, I'm going to put it back on top. So let's make a new tab, and I'm going to put let's just see, 700. Yeah, this is just the data I'm about to send, and nothing's happened yet. Where am I going to put it? I want to put it on top of the one that we know, the 9504. So I need to check that it's expecting XML. It is. I'm going to send that, and fingers crossed. Bad request. Right, okay, that's a bad request because it needs the correct ID. So that's the temporary ID we had before, but we now know it's actually on the server called 9504. So live demos, people, let's put that in there. Yeah, so we're saying we know it's now at 9504. So when we talk about the ID, we've got to say that's got to match. So let's hit send again. And that still didn't work because it's got some junk in there. That's a bit strange. I missed that.
That's the quote.
Yeah, good spot. And yeah, third time lucky. Here we go. So it's got 9504 again. And this time it's telling me it's version 2. So you can see it's the second iteration of this thing. And it should all look the same, but we'll see it's got 700 Main Street. And if I go back to my little viewer and I hit, so that's still saying 681. If I hit display fire again, you can see 700 Main Street. Okay, so that's come through. So that's a basic sort of workflow for this data. You can edit your data in some tool, but of course, remember, you wouldn't really be doing this because this XML will be generated by some, you've got an organization's catalog in your company. You teach your organization's catalog how to export FHIR data. nobody types XML. And then in the input box of your application, you would type in 700, and it would export the XML for you behind the scenes, and it would send it behind the scenes using something like Postman, but behind the scenes. And then whoever you're sending it to gets an update on your data. Yeah, so you get the post, send it the first time, put, once you know which ID it went to, then you can edit it and keep using that one. And then you can use obviously you can use that to fetch it again later. Okay, so we've got about another 20 minutes. Let's move on to our more sophisticated use case of the stability report. So again, here is what you get in your pack. This is a slightly newer flavor of stability report, actually. I just changed it a little bit recently, improved a couple of things. I noticed that every batch was using the same specimen, for instance, which is a mistake. They should use a different one. So I made a couple of tiny updates compared to what's in your on box, but it's all essentially the same thing. So I'm going to post that. This might be a bit more interesting, or by interesting I mean potential to go wrong. So that's 3,000 lines of XML. Yeah, it's a whopper. I don't need an ID because you don't need an ID when you're creating it. I'm going to post it to there. It's still that, and let's see if that works. Go. Good, it did. 201 created, and it's created me another bundle, and this time at 9505. So you can see a pattern there because it was 9504, and it's just put it as the next one on the server. Okay, 9505. So let's see what that looks like. Now I know this does take a long time. This takes about 20 seconds normally, so we'll pull that back. The viewer, when you throw a bunch of XML at the viewer, it chases the links and follows the batches to the organizations and the products and so on. So it does a whole series of querying on the server, and all being well, it'll be back in a few more seconds with something that looks very like what you saw in the HTML view and even maybe in the Word document. Or it might fall flat, but I think it'll be back in a moment. Just in case that doesn't come back. I did come back there, yeah, that's good. Just when I was starting to worry. Yeah, so we've got something that looks like what we expect. So we've got a report, and obviously this viewer is giving some extra sort of information on there to sort of pull. Well, it's the same information, but just tidying it. We've got our diagnostic reports, we've got our performer, we've got our batches, or as we've already seen, and we've got our observations and so on and so forth. And that's kind of what we've already seen. It goes down and down and down. So you can all, well, I guess if you all do it once, it might go very slowly, but that's now on the server at 9505. And what does it have in its data? So this is the data that we posted, and it's got the 0th month in there, and it's got 18 months. And I just didn't bother putting in the other stuff here. I think the idea, is that would have presumably happened. If you're following the protocol, then I guess you had to do those. But it stops at 24. There's nothing at 24 months. So the little scenario that we came up with was to sort of live extend this to add some data in at the 24th month and see if we can make that come up in that slot there. So this is our actual XML for that. Let's make that a bit bigger, not so big that I can't fit it on the screen. So yeah, it's it's a bundle with a diagnostic report in it. This will get a bit techie, but it's got reference to, just scroll down a bit, the results of the diagnostic report are these. So we got our long-term initial on the first sample, on the second sample, the third sample at initial, as in 0 months, and then we've got 18 months and 18 months and 18 months. And then kind of one I prepared earlier, we've got the new data. So as I say, nobody types XML, so this is unrealistic, but I imagine if you had an application, a web, some sort of app, it doesn't have to be a web app, any old app, and it has an Excel-like interface, and you've got the results back from the lab, and maybe they go straight into your database, and you export them. Maybe you have to type them in. If you're unlucky, you have to type them in. You've typed in the 18-month ones, and it generated this XML. Now you've typed in some 24-month results, or you've got them directly from the analyzer, ideally, on some sort of FHIR or HL7 version 2 message feed, who knows. We've got some more data, and so it can be generated in this format. So let's, this is our new data, uncommenting that. So now we've got this 24-month test in there. Don't worry about the name of it. That's just a sort of, human readable type thing. But I know that if I go new data, that connects the new data, which I haven't yet put in, to the report. So the report has now a new section in the report. And what is that? Well, let's change the conclusion. Here's the conclusion. The conclusion said the shelf life can be 18 months. This is a bit of a eat, but let's delete that and go, well, the conclusion is now 4 months. And as I say, nobody would ever edit the XML, but the box in the screen where you type this, you just retype something else. So that's 24 months now. And then let's go down to the next bit, and I would have to actually enter some new data or export some new data. So here it is. We've got an observation for 24 months, and it's a description observation, and it complies with the stated description. And another one, and it's the impurities test and so on, and that has a certain value. And down it goes, and it's 0.2124. So let's go down to the end of that block, and that's now, so that was there. I kind of cheated because that was there already in the XML. I've just uncommented it, so now we have more data in XML. So we have another 100 lines or so, or probably more than that. And then this is a 24-month thing, so it needs a new sample to say, well, this is the sample as it is at 24 months. And what's special about this sample at 24 months? Well, what's special about it is that it was collected in 2024, but it's been stored for 24 months, so it's now December 2022, and it was stored at that temperature. And I'll just put that in, and oh no, yeah, that's right. I'm going to change the actual type of the report as well, just for completeness. I'm going to stop it being an 18-month report, and I'm going to call it month report. And it doesn't look very tidy, but that's okay. And that's where it at the start. Okay, so different data. Let's see if that the server. So where do we want to be? We posted it and it went to line or bundle 9505. So we're going to go to 9505, not 4, and we're going to put that in, the newly edited stuff, and it has to match that. So that has to match, because that's a mistake I made before. And let's just see. This is our newly updated data. I've just edited it, and I'm going to put it on there, and it's that matches, and let's go for it. 200, okay, good stuff. 9505 now has a version 2, and it was right now at 2 o'clock GMT, and yeah, you can't see much, but it's now a 24-month report. So let's go back to our window, and I'll make a new one of those and do it again, and we'll have another 20 seconds to wait until that comes back, and that should be updated. So before it said, well, it didn't say 24 months, it said something else, didn't it? What did it say? Can't remember now. It said 18 months, that's what it said. Yeah, it said 18 months. So that should now come back. I mean, FHIR is normally quicker than this, but the FHIR viewer has to goes to the server in different parts, and every time it finds a resource, it hunts around the server looking for the resources that link to it. So it's doing some extra kind of processing that's that slows it down, because it's an inspection tool rather than a transmission tool. anytime now. Here we go. It now says 24 months. And let's have a look if something's actually happened. And yeah, lo and behold, this line here is filled in. And you'll notice that all the numbers end in 24 because it's 24 months, and then they all end in 18 at 18 months because that's just the way test data goes. And then you could go down the bottom and, you know, I'll go down all the way to the bottom. It probably is all the way at the bottom of the green part. As that. Yeah, so you'll see at the end, hopefully at the end, yeah, you'll see some, oh, that's the accelerated part. So that's even lower. So let's go to this part here. And you can see, well, it's hard to tell this is new, but it is new. You've got the 24-month data, and it's really a bunch of things at 24 months with 24-month values. 0.124 is a real structured value that we saw in the table before. So you can update things like that. Let's do our other scenario was with our substance, and this was a simpler scenario. It was to change one of the test periods. So I interpreted that as being just a change to the plan. So let's bring that, which I don't know if it really is that, but the point is about showing how you change FHIR data really. So the stability report for substance is here. So this is similar to our product one, but it's not on the server. So I'm going to post that to the server. Let's go over there. This is a substance. I'll take my ID out because we don't want that in there. I'm going to just rashly hit send. Did that come back already? That was extremely quick. Yeah, so now we've got 9506. So let's have a look at that one in my browser. So that's one. And while that is cooking, I'll get my other one ready. So hopefully that's going to pop back. But I'm going to change the protocol from being 24 months to 18 months, or 36 months to being 48 months. Yeah. So when this arrives momentarily, hopefully, it will be a 36-month protocol. There it is. Yeah, so we've only got 18 months worth of data, but don't let that put you off. The protocol goes to 36 months. So it's 3, 6, 9, 12, 18, 24, 36. But we're going to change that. We're going to change it to 48 just because that's what we do. And so these are the results. But if I go down to the end in this case, you can see rolling like crazy. You can see that the steps in the plan only go up to 36 months. So we're going to change that. This is the plan rather than the data. We're going to change it to 48. And then this down here will change to be 48. So let's do that. This has got a bit more cutting and pasting, but hopefully I'll get that right. So this is defined as being 36 months. So you can see this is an action at 36 months, and you do this thing called X, which is specified here. But we're not going to do that. I'm going to get rid of that. I'm going to change it to 48 months. And here in the time-honored tradition is one I prepared earlier. Hopefully that doesn't look like it lines up, but hopefully that does line up. And then we're going to put in, yes, there's two phases to this. There's the accelerated and the unaccelerated, the storage one where you just keep the data. Time pass. So I'm going to delete that, and I'm going to put that one in. So a 48-month test in our accelerated bit. we're going to have a new 48 month thing. So this is the original 36 month test. Let's type that one. We're going to get rid of that. A 48 month one. And we're going to, that's it. I think that's it, because we're not actually putting any new data in. We're really only changing the protocol. So I think that is all I need to do, but I'll soon find out if it's wrong. If it's wrong, then the viewer will tell me, which I suppose would illustrate the point of the viewer, that if you do stitch things up wrongly, and it's quite possible I missed something out there, but if I did, I need to change that to be, actually that's not where I want to put it. The put one, which actually says post there, but should change that so it's not so confusing. I'm going to post it to 9506, I think. That's a little bit of our demo. I'm going to put 9506 in there. And it is our stability study. And let me just check it really was 9506. It was. That's not right. Well, it was 9506, wasn't it? That's not the right window, but anyway.
Yes, it was 9506.
It was, yeah, good.
Yeah.
Good. Double check that. And then I'm going to go for that. And it says okay, so that looks good. So it's version, again, it's version 2. So that's good. So I need to find the right window. Oh, here it is. The joys of multiple monitors. So it's at 9506 and it's saying 18 months worth of data, and that won't change. But with a bit of luck, the plan will come back and say that we've changed the sort of protocol. So let's go. Yeah, that's our product one over there. Let's just wait a few seconds for that, and hopefully it's updated the protocol. And if I update one half of the protocol, like the accelerated but not the long term, then my table will come out a bit broken, because it'll probably say one. It'll probably have the wrong number of rows or something. But that's the beauty of the viewer. It kind of it displays your FHIR according to the rules of FHIR. So, you know, if something doesn't match up in your FHIR, the viewer will sort of show that up.
Hey, Rick, this is Mike. May I make a point for the CNC experts in the room?
Yeah.
So what Rick just did is he actually just made a real-time stability update, and he made it not only for FDA, but he made it for the entire world. And I saw that there was a question in there, why use a Cumulus versus the gateway? And this is assuming that Cumulus can build the product pipeline or the data exchange pipeline. But what you just saw him do was do a stability update that did not require a cover letter. It did not require us to repetitively put data into tables or build out Word documents that we then convert to PDF and submit through the gateway. He did it in real time. And now the information can flow seamlessly through the pipeline to the health authorities, and they can review in real time. That's what just happened.
Yeah, cool. I think that sums it up nicely, doesn't it? Yeah. So you can see it came back as 48, and this is the data that was done at this time, and no new data happened at 48 because that wasn't what I was doing. But if we go down to our little, all the way down to our plan, you'll see that our plan part of it has now changed from 24 to 48. So what would happen realistically, you'd have something like this on your screen. You know, you're a user, you're working at a drug company, and you would just go in. I mean, obviously you wouldn't randomly change things, but if you decided to change the protocol to 48, you would just literally update that 48 and all this stuff below would be generated for you. That's kind of how it would work. I mean, we've done it backwards. We're typing the XML and the HTML is coming out, but you'd have something like this, or you'd put, I don't know, you'd maybe put an X in there, and this would be generated, sent to the server, and there would go. You know, you would never be, you know, doing this kind of like stuff, but that's what it would look like behind the scenes. So that's it. That's the end of my bit, really.
Rick,
couple
could you go
minutes
back
before
to
time, I think,
Postman
so.
just for a second?
Yeah, let's go. So here we go. Yeah.
And the pane at the bottom. Oh, there it is. There it is. So
Yeah.
just to note, there's the time, 978 milliseconds.
Yeah.
So just to call that out, that's this time scale we're operating with here. So we're going from, how long does it take to put together an eCTD and send it? We're talking days, hours, weeks, down to milliseconds.
Yeah, that's the idea. I mean, yeah, as I say, this viewer takes about 20 seconds, but that's just a kind of debugging tool that does lots of checks in the background. But yeah, in theory, you could change that to 96, hit the send button. Postman would be triggered sort of behind the scenes. It wouldn't really be Postman. It'd be the same features that Postman does, but behind the scenes. And yeah, 978 milliseconds later, or depends on your internet connection and all the rest of it. And this is going to. I'm in the UK. And this is going off to somewhere in the States, so it's gone across the Atlantic in under a second. So that's not too bad, is it? Okay, that's my little scenario for now. Are we doing a break now, or do we do questions now? So perhaps it's break time.
Yeah, I think we're right on time for the break, Rick. So thanks so much for doing the demo and going through all that. So I think
Sure.
when we come back, we can go into sort of the discussion portion of today's testing event. So I think we should start the break now, and then we should come back at 10:30 and be ready to discuss.
All right.
Sounds good.
All right. Thanks, everyone.